So Alex is here. He's going to tell you a little bit about biotechnology. So it's all yours.
Alright folks, my purpose here today is to give you a small taste of what biotechnology is. Biotechnology is a very important topic because it covers medical, agricultural, engineering, any kind of science. It's linked into biotechnology. All the way from the ancient times to present and beyond, biotechnology has been used. They may have not known they were using biotechnology, but they have been. So what is biotechnology? I take this quote from National Health Museum. It's the purest form. The term biotechnology refers to use of living organisms or their products to modify human health and the human environment. So basically, you're putting living things to help us live better. Got a little bit of history here for you. Before the 20th century, the first people to use biotechnology were the Sumerians. They brewed beer. Yeast is living bacteria, so when you inject it and ferment alcohol, you're using biotechnology. Like I said, they didn't know they were using biotechnology, but those were the first people to use it. The first insecticide use was used by the Chinese in 100 BC, and what they would do was grind up chrysanthemum, which is a flower, and spread it on their crops, and it would prevent insects from attacking it. In 1675, Antony van Leeuwenhoek discovers protozoa and bacteria. Now bacteria is the fundamental thing that all biotechnology forms around, is using bacteria for changing bad bacteria into good bacteria that we can use for our systems. In 1797, Edward Jenner inoculates a child with a vaccine to prevent smallpox. This was the first use of a vaccine, and it was effective. It's the same way that we use them today, except this was way back when. In 1855, what I consider the most important step in biotechnology was discovered because E. coli bacteria was. E. coli bacteria is what biotechnologists use to synthesize everything else. They use it as a modification and a tool to study the background of other bacteria, as well as turn that bacteria into good bacteria that we can use for ourselves. And I'll show you an example a little bit later. In 1863, Gregory Mendel, he did his pea study and found out that the traits are genetically passed on from the parents, and that's another step towards genetics, but it's also biotechnology because it allowed us to insert genes later on, and we can control what we get back out for a product. 1920, HGH was discovered, human growth hormone. It's used right now for if kids have growth deficiencies, they can inject HGH, and it will prevent an increased growth of the certain area, whatever they're trying to grow. They've also recently used it in preventing aging. I don't know where that's going to go, but that's just a new topic they've been working on, and for weight loss actually. 1928, penicillin was discovered. All of you guys know what the importance of penicillin was. The first antibiotic, still used today in some formats. 1973, another very important step, was discovering recombinant DNA. Now, what recombinant DNA is a hybrid DNA. They take a piece of something else that they want to code for in a new piece of DNA, they put that together with another thing, put it inside a bacteria or something, and then they can use that to manufacture lots of different proteins, and then you can use those proteins for human health. There's also many uses for recombinant DNA also. 1981, first genetically engineered plants, and the first mice were cloned actually. It's also a step of biotechnology. 1982, another very important step, was Humulin was created by a company, and it was created by using recombinant DNA, and they would make insulin out of it for people who are diabetic, so you can make more insulin and make it more affordable for everyone else. 1983, PCR technology was developed. PCR stands for polymers chain reaction. It allows a biotechnologist to take a molecule and literally make dozens and dozens and dozens of copies of it using enzymes and heat. So you have unlimited number of copies you can work with. Otherwise, this stuff would take, forever to develop because you wouldn't have the copies you needed. This sped up biotechnology tenfold at least. 1984, DNA fingerprinting was developed, used for crime scenes, finding your genetic parents, all that stuff. 1993, FDA approves bioengineered foods, which we now have in the market all over the world today. And 1997, Dolly the sheep was born. It was the first cloned sheep that actually, lived for quite a long time. Lived to 2003, I believe. And that was a major step, like I said, towards genetics, which is a derivative of biotechnology. And in 2000, the rough draft of the Human Genome Project was done. Now they worked on that for over 12 years after they got federal funding for it. What the Human Genome Project does for us is allows us to, we can see all the genes in our human, so we can take good genes and splice them into, say, if someone had cancer, and you could eliminate the genetic probability of getting cancer by taking out those cancer cells and putting the new ones back in. That's just one example. There's hundreds of examples. Biotechnology in medicine. The graph over here shows how they make insulin, actually. Whereas you have your pancreas, which makes natural insulin, obviously. Well, then you can take what you want from there, splice it into a plasmid, put that into a bacteria, usually use E. coli for that, which is normally a bad bacteria. And then when that makes copies of itself, it copies that over and over and over and over. So therefore you get tons of insulin you can use, all from just one segment of DNA originally. You can also use it for gene therapy, which would be splicing out bad genes, putting good genes in. Prevent, like I said, cancers, cystic fibrosis, multiple sclerosis, any of those genetic defects. They can also produce organs for transplants, and there's two ways they could do that. They can grow them in pigs. People have done that before. And so you could grow organs in pigs, like the heart or something, and transplant it. That's been done a couple times, very successfully. And they're also working on growing them in, actually, like dishes. They can actually grow tissues and eventually organs that they can transplant into you. That's controversial and all, but that's a completely different subject. And like I said, a real-world example of biotechnology in medicine would have been the manufacture of insulin. Another big area of biotechnology would be in agriculture. Like I said, they use biotechnology in agriculture as early as 100 BC in China to prevent insect damage. It's also used to improve appearances, taste, shelf life. It can prevent freezing. It can help with droughts and floods to make them become resistant. You can start growing stuff that would take a lot of rainfall in less rainfall areas, and therefore you can create unlimited amount of food if you just start working towards that. You can also increase meat production on beef cattle, or dairy production on milk cattle, dairy cattle. A real-world example of this would be the golden rice. And for the Asian countries, this is very important. Rice is a very important part of their diet. So what they did was they genetically engineered golden rice. One of the steps they did was to add vitamin A to it, which prevented blindness, which was a big problem in Asia and still is. So if they started feeding them rice with vitamin A in them, then they would prevent the blindness from occurring. They also used it to, they spiced in some stuff that would prevent bacteria growth that would kill enough food per year to feed 60 million people. So you save 60 million people by just splicing in good DNA and preventing crops from being damaged by bacteria and insects and stuff. And that's actually it.
Interesting. Be a lot of good jobs.